Okay, good morning everybody. Let's get started so we don't get too delayed. My name is Enrique Martín Gallo from Universidad Autónoma de Madrid, and I'm here with Julia García Alvarado from IRSI Caixa. We will be moderating this MESA REDONDA BÁSICA 9. And today we have outstanding speakers who will be presenting some of their work on HIV cure, implication of tissue immunology, and also pediatric HIV. So let's get started with our first speaker. Welcome Nancie Archin. Her lab is focused on latency reversal regulation, the influence of sex, and also epigenetics. And she's located at North Carolina University. So thank you so much for accepting the invitation. And the title of her talk will be HIV Curing Women, What We Know and Where Do We Go From There. Thank you.
Thank you very much. Buenas tardes. Thank you to the organizers and particularly Maria Salgado for the invitation to participate as a speaker in this conference. And it's a pleasure to be here, and I'm looking forward to maybe more interaction with some of you later. Now, today I'll be giving a general overview of what is known about the HIV reservoir in women and how that might impact cure approaches. And at the end, I'll have some thoughts of basically where do we go from here, which is the title, part of the title of the talk. Let's see. Perfect. So as you heard yesterday beautifully from Javier, while there is the major obstacle to, so while antiretroviral therapy can suppress HIV infection, it is not a cure, and in the absence of therapy, virus will rebound. And this is because of these populations of latently infected cells that contain replication-competent viruses that persist for years on ART. There are no known extracellular markers associated with latency. Latency is established early during an infection, and it's not recognized, the infected cells are not recognized by the immune system. And while with the, sorry, I have to look at my notes. And while with increasing access to effective HIV prevention, diagnosis, treatment, and care, HIV infection has become a manageable chronic health condition. However, a cure is desperately needed as HIV is a lifelong infection, and a significant number of people are still do not have access to antiretroviral therapy. And as shown here in this slide, what I'm showing you is that especially that half of the people living with HIV infection are women, and women are traditionally underrepresented in HIV cure research. So women constitute, as I mentioned, half of the people living with HIV disease, and this study published a couple of years ago basically showed that on average only about 11% of people women are recruited to HIV cure research. So the implication is that most of what we know about the HIV reservoir comes from studies involving men. So a clear understanding of the nature of the reservoir in all people, however, is necessary to help guide cure interventions. And there are many biological factors that may affect HIV persistence in the reservoir in women. Women are more likely to be spontaneous controllers of HIV. Women are overrepresented in most post-treatment control cohorts. Women are reported to have lower levels of plasma viremia and higher CD4 T cell count than men. Untreated women, for instance, exhibit levels of viremia that are up to 40% lower than men. However, women tend to progress faster to AIDS than men. And this is thought to be because of that women have higher levels of chronic immune activation compared to men for a given viral load. Sex hormones can also influence the reservoir. During active infection, HIV RNA levels were shown to vary with the menstrual cycle in the female genital tract. Lower plasma viral loads were observed during the secretory phase of the menstrual cycle, and this is the stage where estrogen levels are at their peak. And in addition, there have been studies showing that under estrogenic conditions, estrogen can complex with estrogen can cause the estrogen receptor to complex with beta-catenin at the HIV promoter to suppress HIV transcription. So in order to gain a better understanding of sex differences in HIV reservoir size, we performed a small cross-sectional study where 22 ART suppressed HIV seropositive women were matched 1-to-1 with ART-treated men. And here, what I'm showing you is the study population characteristics. And as you can see, it's pretty balanced. And what we did in this study, this was a very small study, we compared the reservoir of the 22 women matched to the 22 men. We also compared the reservoir from the 22 women not matched to all the men. And we looked at both replication-competent HIV as performed by the Cuvora assay. And the Cuvora assay essentially is an outgrowth assay where you take CD4 T cells and co-culture them with PBMC blast to expand what virus coming out of the cells, and then you use Poisson statistics to estimate the size of the reservoir. But as you can see in both, compared to the matched men here versus the women, we didn't really see, although we saw perhaps a trend towards lower reservoir in the women compared to men. However, this did not reach statistical significance. So there is no significant difference in the frequency of replication-competent HIV in women versus men in this small study that we did. We also look at, because the Cuvora can underestimate the size of the reservoir, we also look at the IPDA, which is the intact proviral DNA assay, just look at proviruses that are more likely to be intact. And as shown here, we also did not see, we saw similar frequencies of intact and defective proviruses in women when compared to men. Now, in a different study, and this is reported from Eileen Scully, what they did, they compared 26 men and 26 women, and they matched them on more than we did. We just matched, when we did our study, we matched it on age and race. They matched on suppression, CD4 count, and NAD CD4. And like us, they did not see a difference in the total reservoir, here shown looking at the DNA reservoir. However, they did see that women tended to have perhaps smaller multi-splice HIV RNA. Also, they also saw that women had lower residual plasma viremia. And at the bottom here, they also did an inducible assay just to look at the inducible reservoir. And what they saw that perhaps women also do have a smaller inducible reservoir compared to men. So Jessica Croger and her group studied 57 women from in her REDONDA cohort and compared it to 33 men. And here what they're doing is that they're looking at both the replication-competent reservoir, again by doing the Cuvora, and looking at total DNA. And what they saw again is that women tended to have a smaller inducible reservoir compared to men, but no clear differences when they looked at total HIV DNA. So in this very interesting cohort, this is not actually looking, this is not looking at reservoir size, but what Sarah Gianella and her group did here is just looking at RNA positive cells as they follow women from the time when they were premenopausal, like meaning during their reproductive age. The same people were followed all the way up to when they become postmenopausal. And they saw this interesting observation where the reservoir also, or I should say cells that are easily inducible to produce virus were more frequent when so basically they were able to reactivate HIV better when the women were postmenopausal compared to when they were premenopausal, suggesting that perhaps in an estrogenic environment, the reservoir could be more difficult to induce. So it's more difficult to induce in premenopausal women compared to postmenopausal women. So again, suggesting that perhaps estrogen is having an effect in the ability to induce the HIV reservoir. And this is very interesting because this is recent data from John Miller's group and Raj Gandhi, when they followed people over time, looking at decay of the reservoir, and they saw three patterns. In the first pattern, you had this rapid decay followed by slower decay. And in the second pattern, you had a decay, but then everything plateaued. And in the third pattern they saw was you had a decay, then you saw an increase of the reservoir. And this was really interesting because where they saw the increase was in two postmenopausal women. And they think that what's happening is that in perhaps in women, you're getting this expansion of the reservoir. So what this increase could be coming from large clones of cells containing provirus in them that has been expanded in vivo. But again, suggesting that perhaps as women become postmenopausal, they have this the less effect of the estrogen on the reservoir. So women tend to have a more robust type 1 interferon response. For instance, in some studies, it has been shown that plasmacytoid dendritic cells and ISG induction in women are higher when the cells are stimulated with TLR7 ligands or with inactivated HIV-1. And it's believed that this larger and more robust interferon alpha, type 1 interferon response might explain some of the differences in the clinical manifestation of HIV infection between women and men, including the better control of HIV viremia by women during primary infection. It's also suggested that estrogen can enhance the ability of plasmacytoid dendritic cells to respond to TLR7 stimulation. So we wanted to determine if there are sex differences in interferon stimulating gene expression during treated infection in women by looking at the expression of various interferon genes in our cohort of women. And while we saw some variability between men and women, what stood out to us really was ISG15, where we saw that even though these women had been on antiretroviral for many, many years, there was a higher expression of ISD15 in CD4 T cells of women compared to men. And this was very interesting because recently, I have to say that when we looked to see if there is a direct correlation with the high level of ISD15 expression and the reservoir in women, we did not see a correlation. However, in this most recent study, what Thielin did is that they took PBMCs and they stimulated them with TLR7 and 8 agonists, and what they saw is that the percentage of plasmacytoid dendritic cells expressing interferon alpha was significantly correlated with the reservoir. So basically, the higher the amount of interferon alpha that was being produced by the PDCs upon stimulation by TLR7 and 8, the lower the reservoir. And they also saw an influence of ISG15. Essentially, the ISG15 mRNA correlated significantly with the ability of TLR7 induced production of both interferon alpha and TNF alpha, shown here at the bottom of the graph. So basically, the more ISG15 you produce, the more likely your PDCs are able to respond to TLR7 and TLR8 and produce more interferon alpha. Again, suggesting that perhaps there is something there with women being able to mount a robust immune response, type 1 immune response, type 1 interferon alpha immune response, and be able to control the size of the reservoir. So overall, looking at multiple studies, looking at the reservoirs, there's been some mixed finding across different studies. Most of the studies finding a significant difference in reservoir size between men and women typically show about only a twofold difference in persistent HIV frequencies between men and women. We have yet to try to understand what the biological implications are and how that might affect cure approaches. One thing many of us have been interested in is, since a lot of the difference, most of the difference that we see in a reservoir between men and women seems to come from the ability to induce the reservoir, so basically the inducible reservoir. We wanted to know, are there differences in a proviral landscape that contributes to differences in the inducibility of the reservoir between men and women? And then so we did this quick study where we used this assay we call HiveSeq, using nanopore sequencing to look at full-length sequences in both women and men and compared to men and women. And this is qualitative data and not quantitative at all. But what you can appreciate is, if you go across all the blue line here, these would be, if you see a complete blue line, those are your intact proviruses, and any time you have white, it means you have defective proviruses. You can appreciate with this just very simple study is that it looks like women tended to have, we tended to recover sequences that was mostly defective in women compared to men. Now, in probably a more in-depth study performed recently by Ton Singh and Matthias Lechterfeld and Shiruyu's lab, they used their FLIPS-seq and MIPS-seq assay to look at near full-length viruses, as well as to perform integration site analysis. And what they saw was that there was no difference in the reservoir, in the intact reservoir and the defective proviruses between men and women. But as expected, the reservoir in elite controllers was smaller, but people on ART, let me go back, there was essentially no differences between the men and women. But the women tended to perhaps have a higher level of intact proviruses. And the open circles mean it's below the limit of detection. So, but they made these really interesting observations when they did sequence analysis, and what they noticed is that most of the reservoir, although the women tended to have more higher levels of intact proviruses, what they noticed was that most of these intact proviruses were clonal. Again, going back to the clonal story, that in women, although there are perhaps more intact proviruses, they tend to be very clonal. And interestingly enough, most of these clones tend to reside in areas of the genome that are harder to reactivate. So basically in centromeres, the same fingers, as opposed to the genic region. When you compare men versus women, most of the reservoir in the women tended to be in this harder to reactivate regions of the genome. And this was true for the intact proviruses, but not for the defective proviruses. And what they've hypothesized is that perhaps because of the more robust immune response of women, the viruses that once resided in the genic regions, as they reactivate, they get cleared by the more robust immune response. So selecting for viruses that tend to be, enriching for viruses that tend to be more in the areas that are harder to reactivate. So the last point I want to touch in regarding the reservoir in women and cure approaches is different types of cure approaches have been studied for clearing HIV infection and depleting the reservoir. And one of them that's gained a lot of steam is called the shock and reverse, or shock and kill approach. Basically, the idea here is you treat cells with latently infected cells with latency reversal agents. hopefully to reactivate the virus from latency, and then you can use some immune function, augment the immune function to block and kill the infected cells, and you would use R to block new cells from getting infected. And by augmenting the immune function, you can clear the infected cells that are now making, that are now producing antigen. But as I mentioned, estrogen tend to be very suppressive of HIV transcription. As I mentioned, beta-catenin can complex with the estrogen receptor at the promoter to restrict HIV transcription, and estrogen can also blunt TCR reactivation of latent HIV infected CD4 T cells ex vivo. And this was a study done by Jonathan Kahn a couple of years ago, and what he's shown basically here, if you compare cells from women in red, this is his if you compare cells from women here in red and in men in black, if you try to reactivate these CD4 T cells by stimulating the T cell receptor, and you can see you get robust reactivation in both men and women. However, in the presence of estrogen, you can see that estrogen completely blunt your ability to reactivate HIV from latency, and this was more pronounced in women compared to men. However, if they take something that inhibits estrogen, for instance, ICI, what they saw, if they treated the cells with latency reversal agents such as SAHA, in the presence of inhibiting the estrogen receptor, you get more robust HIV reactivation when you put something to block the estrogen receptor. And this was more pronounced again in men versus women. Sorry, this was more pronounced in women compared to men. So this study here led to this ACTG study that was done a couple of years ago now, where basically women were given tamoxifen every single day for about 38 days. And then they were given doses of vorinostat. And the idea here was that if tamoxifen is blocking the estrogen receptor, you would hopefully get a robust increase in cell-associated RNA, meaning that the reservoir is being induced. And they looked at this by looking at cell-associated RNA in CD4 T cells and also in using the EDITS assay. So basically two different assays to see if the combination of estrogen really, the combination of tamoxifen and vorinostat augmented the ability to induce HIV from latency in vivo in women. And this was, and although, as you can see above here, they did not, so modulating the estrogen receptor of tamoxifen did not augment vorinostat-induced HIV RNA expression in postmenopause women. So this was a bit of a disappointment, but it was at the same time, it was a very important study because this was actually the first field trial done exclusively in women. And additionally, one of the reasons perhaps this didn't work is because due to the cancer causing abilities of tamoxifen, the study was restricted to only looking at postmenopausal women because tamoxifen can be both an estrogen agonist and an estrogen antagonist. And then so the study had to be restricted to postmenopausal women. And perhaps in postmenopausal women, the activity of the activity of the estrogen is the ability to induce HIV latency, as I mentioned earlier, is less restricted by estrogen in this environment. So in summary, I hope I have convinced you that sex differences in HIV persistence is a complex with mixed reports of both evident or no evident differences in HIV reservoir frequencies as measured by either when you look at the replication competent virus or when you look at IPDA, just looking at the intact virus, or GAG DNA by PCR in ART-suppressed seropositive men and women in both match and unmatched analysis. Recent studies indicate that estrogenic environment may detect the inducibility of the HIV reservoir ex vivo, so basically being able to better reactivate the reservoir in premenopausal versus postmenopausal women. The reservoir in women may be composed of intact viruses that have largely expanded clonally, and they tend to have a more lock and block features. Again, when looking at future HIV cure that are using LRAs to reverse latency, the reservoir may be more difficult to reactivate in women. The suppressive effect of estrogen and HIV expression, therefore, may confound the interpretation of anti-latency therapeutic trials. And of course, robust type 1 interferon responses in women may be associated with a lower intact reservoir. Studies with large cohorts of men and women, therefore, to help better define the biological relevance of the reservoir differences and the implication on cure interventions, I think are needed. We also need well-controlled, longitudinal studies of women from reproductive to postmenopausal age to better understand hormonal influences on reservoir dynamics and how these can affect cure approaches. What is understudied is the effect of feminizing hormone therapy on the reservoir in transgender women, for instance. How does hormone therapy impact proposed cure approaches? We also need to include more women, both cisgender and transgender, in cure clinical trials. This may involve, for instance, designing trials with specific needs of cisgender and transgender women in mind to increase participation. For instance, can we provide child care when a woman comes to the clinic to participate in a study, to encourage women to participate in cure-related research. And so to conclude, we know at least 4 women have been reported to be cured of HIV. The New York participant who received transplantation of banked umbilical cord blood with Delta 32 mutation, CCR5, and also stem cells from bone marrow of an adult relative. The other 3 women, however, the Esperanza participant, a Barcelona participant, whom I'm sure you're all familiar with, and Lorraine Willenberg from California, are all able to control HIV without antiretroviral therapy and without stem cell transplantation. And as I mentioned, women in general are reported to have a more robust immune response to pathogens, so we do need more studies to understand the role of the immune response in sustaining a functional cure in these women. And perhaps the lesson learned could potentially help us design more efficacious cure interventions that are not only beneficial to men, but also beneficial to to all people. And with that, I want to thank some members of my laboratory who were involved in that work and all our collaborators and funding, and also the participants without whom we could not, these studies would not have been able to be done. And with that, I'll stop and thank you very much. And I think I'm supposed to take questions right now.
Yes, we have time for 1 or 2 questions. Pepe?
Thank you, Nancie. Very nice talk. In addition to concerning your last slide, with these 4 women that have been reported as potentially cured, the experience with many cohorts of lead controllers and long-term non-progressors is that up to 50% of these patients are women.
Right,
so
right.
Which hypothesis would you favor to this status of lead control? Is
Yeah.
it interferon, natural immunity, more adaptive immunity? What's your...
Yeah, no, it's a very good point. And I think I mentioned that women are overrepresented in post-treatment controllers and elite controllers. And I think, again, it comes back to the immune response. I kind of favor the innate immune response because I think we're learning more and more about the ability of women to mount these type of responses and their influences on the HIV reservoir. So, and then also with the elite controllers too, I like the hypothesis that perhaps Matthias is proposing that perhaps, again, because of this robust immune response, you're just getting rid of the viruses that are easy to reactivate. And then what you end up getting is like enriching for viruses that perhaps are in areas that are harder to reactivate, like in the lock and block areas of the genome.
José Miró, brilliant presentation. My question is the integration sites between men and women. It seems different, so women have more inactive sites.
Yeah.
This can be the reason why they are more elite controllers.
Yeah, that's what I
also,
just, yeah.
Similar. And also the difficulties to find a cure in this population. So if you cannot reactivate the virus, how the immune system will destroy the infected cells?
Well, it's possible that it's in such an area that, like especially if you end up in a centromere, perhaps you don't need to activate it because there's no way it's ever going to come out. Right? So,
thank you.
Yeah.
Thank you. Great presentation. So I was wondering about your analysis from nanopore sequencing. Did you check, or do you have the idea of how what type of deleterious viruses you are having, and if you saw, for example, maybe higher ApoB mutations, if your hypothesis is that interferon is maybe higher, and then or higher diversification, or the opposite, maybe lower diversity that leads to to worse viruses. And the second, I just have you, or do you have plans to look at any tissues that could be very interesting for this thing?
Yeah, no, that's a very, very good question. Unfortunately, with the type of sequencing we did, we couldn't delve deeper into that with the nanopore. It was just basically qualitative, whether or not we couldn't tell you like what the percentages were. Regarding the tissue, right now we are very interested in looking at the reservoir in different tissues, especially in the gut. We have a protocol to try to look at different areas of the gut to better understand if there is a correlation with the reservoir in the gut versus the reservoir in the blood, and what cell types in the blood. So we have some single-cell RNA-seq, as shown in a previous talk, that shows like a lot of the gut reservoir is coming from the resident memory T cells. So it's something we are very interested in.
Okay. Quick question, Maria.
Yeah. Thank you, Nancie. Very, very nice presentation. I have two small questions. One is if you saw some study, because you saw that the reservoir is not that different, but then the reactivation is different. So now there are a lot of treatment interruption studies. If there are some difference in the time to rebound of those viruses, if there is a delay with the woman.
Yeah, unfortunately they haven't. Yeah,
we
need to recruit.
Very interesting. And also the second is just more.
You guys should do that.
and the second would be more related, like maybe a personalized medicine where maybe latency reactivators agents are more maybe for men, but for women maybe we're thinking more, I should say, block and lock, right? Because it's more difficult to.
Because they're already halfway there. Or it could be that we need to find different latency reversals that can target more the viruses that are harder to reactivate in women.
Yeah, thank you very much.